Biology Part 193. In dye-neutral plots, the photoperiod is unimportant and these other factors become main external influence of affecting flowering time. Many tropical plants and some temperate species including dandelions, sunflowers and tomatoes are dye-neutral. The measurement of the photoperiod by short dye and long dye plot is now known to depend on a light sensitive pigment called the phytochrome, which is present in the leaves. The phytochrome exists in two forms, each of which can be converted to the other by light of appropriate wavelength. The inactive form of the phytochrome is converted to the active form following the exposure of the red light with the peak sensitivity at 660 nm. PFR is slowly converted back to the inactive form in the darkness, may be rapidly converted to PR by exposure of far red light with peak sensitivity at 730 nm. Well, the daylight is a mixture of wavelengths containing both red and far red light, but red wavelength predominant and the red wavelength predominates, so that during the day PR is converted to PFR, which accumulates in the leaves. The onset of the flowering seems to be determined by the balance between the two forms of phytochromes pigment, and which way some switches on the genes responsible for flowering. Well, flowering may also be initiated by hormones transported from place to place around the plot. I beg your pardon for sneezing in front of the camera, but that's uncontrollable, of course, unintentional, true. I beg your pardon again. Flowering may also be initiated by hormones transported from place to place around the plant. The experiment of the figure provides clear evidence for some type of the chemical influence in two cockleburr plants separated by light type partition have been grown with different photoperiods. Cockleburr is a short dye plant with a light with a critical around 15.5 hours with a 12-hour light per day. Flowers are produced with 18 hours of light. Flowering is inhibited. In B, flowering branch from 12 hours plant has been grafted on the 18-hour plant through a hole in the light tight partition. If the two parts of the plant continue to receive 12 or 18 hours of light respectively, the 18 hours plant subsequently produces flowers, although it would not normally have done so. Experiments of this sort have been taken as evidence for the existence of the special flowering hormone named as the florigen. However, such a substance has never isolated, and nothing at all is known about its possible chemical composition. Indeed, it seems more likely that the observed is due to interaction of two or more hormones already described. The whole range of the other light-dependent plants responses involves the photochrome as the photoreceptor molecules. These includes the germination of some seeds and the normal development of seedlings, leaf apposition, and of course the fruit formation. The effect of the far red light seems curious in view of the normal spectral composition of the daylight. However, light filtering to the flower of a forest contains a greater proportion of the far red light, which is absorbed less effectively by leaves. Consequently, it seems possible that the far red conversion may be functional and adaptive to some types of responses which are not yet understood. So I'll tell you more about it, and I'll talk about the nastic movements in the plants. So that's what we'll do in the next video. Right now we're putting an end to this video. We'll discuss the Nasdaq video in next video. We'll discuss the Nasdaq movements in next video.